So, good morning, good afternoon, and good evening. It is my pleasure to welcome you to the fourth session on advances in enhancing the microbiology safety of food. The views expressed in the following four presentations are solely those of the speakers and do not necessarily represent the view of the International Life Sciences Institute. Each speaker had the total freedom to prepare her and his presentation. It is now my pleasure to welcome Dr. Mike Doyle. He is a retired Regents Professor at the University of Georgia. He has published more than 500 scientific papers. It will be the chair of today's session. And he is also the co-chair of ILSI Global and the chair of the organizing committee who put together the program of this event. So, Dr. Doyle, I'm yielding to you.
Thank you very much, Stefan. Am I coming through loud and clear?
Yes.
Okay, thank you. Now, I'm really excited about today's session because we really have a star-studded lineup of scientists who are at the cutting edge of research that address the microbiological safety of foods. Whole genome sequencing has revolutionized the field of foodborne disease surveillance and detection. And today we're going to learn about the application and new developments of whole genome sequencing and next generation sequencing as it relates to foodborne outbreak detection and surveillance. And also, although COVID is not a foodborne disease, it's a respiratory disease, it certainly has adversely affected the food industry's workforce. And today we're going to learn about this and what measures are most effective in reducing COVID risk to the workforce. So you see here our lineup of stars who are at the cutting edge of research in these areas. And we're going to begin with Kelly Heiss, who is the chief of the PulseNet Outbreak Detection and Surveillance Unit at the Centers for Disease Control. PulseNet has been with us for decades, and it's been very effective as a fingerprinting method to help detect outbreaks. But whole genome sequencing, which was introduced about 2 years ago into the PulseNet system, has brought this surveillance and detection to a whole new level. So Kelly is going to enlighten us and share with us what's been happening at CDC in this area of whole genome sequencing to detect foodborne outbreaks. Kelly?
Hi, everyone. I just want to thank Dr. Doyle and the committee for inviting me to speak today. Hopefully you can see my slide. It looks like you can. So I'm just going to go through detecting foodborne disease outbreaks through using whole genome sequencing, and then some of the tools that we utilize. I'm going to start with an introduction to PulseNet. I realize not everyone may be familiar with PulseNet. If you are, this will be a refresher. Okay, so what is PulseNet? We're a national network. I'm going to specifically talk about PulseNet USA, although I will give a brief introduction into what PulseNet International is. We're a national network of 82 state and local public health, food, and regulatory agency laboratories. And we're all coordinated by CDC and APHL. We detect foodborne disease case clusters that may become larger outbreaks, provide real-time molecular surveillance of the most important bacterial foodborne diseases. These include STEC, Salmonella, Shigella, Listeria, Campylobacter, and Vibrio. We collaborate with our foodborne epidemiologists in investigating these outbreaks. And some of the ways we do this are by separating out outbreak-associated cases from sporadic cases, providing case definition, and assist in rapidly identifying sources of outbreaks. And overall, as a network, we act as a rapid and effective means of communication between the public health laboratories. So this is a map of the US. We have the different colors. Each color represents a region, and you'll notice a star in each of the regions, and these are our area laboratories. So our area laboratories help provide troubleshooting assistance, surge capacity testing, as well as some other things for their the labs in their region. And this just lets you know that every state is involved in PulseNet along with Puerto Rico, and we have also several local city and county labs that are involved as well. So this is PulseNet International, divided into 7 regions in 89 countries. As far as where PulseNet International is, USA, Canada, and Europe have fully transitioned to using WGS for PulseNet surveillance activities. But other regions, the situation is more variable. Some are just starting PulseNet activities for the first time. Others are exclusively using PFGE, which was an older subtyping method that we used to use. And a few are preparing to make the switch from PFGE to WGS. Some key priorities in the PulseNet International Network are offering technical assistance, resources, and training for partners, facilitating communication, data sharing, and regular meetings, and developing short and long-term visions for global PulseNet surveillance activities. So there's basically three basic elements of PulseNet. You have your data acquisition, so that's a picture of a MySeq instrument that produces the data that goes into our system. We use bionumerics as a software analysis system, and all this information is shared with epidemiologists in CEDRIC. CEDRIC is the System for Enteric Disease Response, Investigation, and Coordination. It pulls in data from the PulseNet national databases into the system on an hourly basis. What this allows for is direct communication with our epidemiologists without them having to have the software system, the bionumeric system. And then another way of exchanging data is through the PulseNet SharePoint site. And that's just where we post comments about ongoing investigations, as well as troubleshooting and SOPs. So this just kind of shows you a little bit of how the network works. So in each individual laboratory, PulseNet laboratory, they do their actual sequencing there, and they analyze the data, and then they upload that information to our national databases at CDC in Atlanta, Georgia. And what they're able to do there is they're also able to pull back information into their local databases so they can do local cluster detection. They're also able to connect to the national databases and see what's going on at the national level. And then we are able to do national cluster detection and looking at the data on a daily basis. So this may look a little complicated. It's really not. It just kind of breaks down what all is involved with our data analysis workflow, just so you can kind of get an idea of how complicated it can be. So you have, again, you have your MySeq, you have your raw sequence data at the public health lab that's produced. All the information is stored somewhere. We don't require the states to store them anywhere except for NCBI, but they will store it on base space or their cloud storage, or maybe even an external hard drive at their local location. That sequence data goes into a reference ID database. A reference ID database is where you just put all your sequence data. It doesn't matter if it's Salmonella, E. coli, Listeria, all goes in this one database. They submit their data to our CDC's calculation engine, and what they get back is species identification and an initial quality check for contamination. They verify that, and then what it does is it pieces it out. So all your Listerias go here, your Estec, your Salmonellas, and it knows which organism-specific database to go to. So my Salmonellas are going to go to my organism-specific database, and then once they're in there, they submit to the calculation engine again, and what they get back are their allele calls and genotyping results. And genotyping results will be serotype, AST data, virulence, and other information. They're going to verify their quality one more time, and then when we ask them to upload this information to the national database within 24 hours of results. And then they can perform surveillance in their local database. You'll also see that we have them submit their raw sequence data to NCBI for either storage or just so that those outside the PulseNet network have access to the data. Okay, so now there's a brief intro of PulseNet. We're going to get into whole genome sequencing and allele codes. So I decided to throw this slide in because I realized I was talking about all this stuff with alleles and didn't really explain what that was. So when you push your raw sequence data information to the calculation engine, what you get back are allele codes at different loci. And you'll see that this is actually an example of a Listeria cluster in our national database. So the allele calls at each locus are basically compared within each other, and you get thousands of these. So obviously you're not doing this by hand. You need an analysis system to do this, and that's what we use bionumerics for. And it will, if you pull all these into one window, it pulls up a phylogenetic tree, and it will tell you where those differences lie. So those differences go into the calculation or creation of allele codes. These are also used when we are doing cluster detection, and we look at these allele differences to determine if what we see is potentially a cluster worth investigating. So allele codes. So it's just a code, and I'm going to break that code down for you in another slide. But it's assigned to every sequence that passes quality in the PulseNet national databases. And these can be downloaded back into the local databases. What this allows for is then you now have a number or a code that you can search on in the whole database without having to do all these calculations over and over and over again. Allele codes provide a hierarchical name to show relatedness between isolates, and these are based off of the core genome, or CGMLST. They offer a compact view of the entire population structure for an organism database, such as Salmonella or E. coli. Therefore, you need to think about these allele codes in terms of population and not outbreaks. And remember that allele codes are not the same as outbreak codes. So super busy slide, and I thought about just breaking this into two. What we can do is start from the top and work down. So the top is a sample breakdown of an allele code. The first part will tell you the organism. So this is LMO. So that tells me it's Listeria monocytogenes. The version is 1.1. So we've already had a change. So it went from 1.0 to 1.1. And then each number, as you go further into the allele code, you'll notice the number of alleles goes down. And this just means, so if you're looking at your example here, sequence A and sequence B, they match up to the third number. So that just means that sequences A and B are between approximately, there's something popping up on, approximately 36 and 19 alleles of each other. And you can see that up at the top in the code where the third digit, if they're at a third digit and they don't have the fourth digit, they're somewhere in between those two. They're not exact, but it's just a tool that gets you closer to seeing which ones are more alike. And then we have, I have a table that just kind of summarizes this for Campylobacter, Aztec, Listeria, and Salmonella. The number that is highlighted in purple is the digit in which we recommend local users to do their cluster searches on. So for Campy, it's the fourth digit. Same for Aztec, same for Salmonella. For Listeria, it's actually the fifth digit. This is a tool that's in our bionumerics software. And you can see up at the top, you just choose which digit you want to look at. And like we just said, we're going to look at the fourth digit for Salmonella. So I'll do 4 digits. I can decide to include or not include non-human sources as well as historical isolates. I will say for initial cluster detection, we usually don't include those at first. We identify our cluster size. We typically say that you should probably have 3 or more in your cluster. And then how many days do we want to look back? So we typically look back 60 days. In smaller states, maybe they want to look back further. And then your result set, when you hit recalculate, you'll see the allele code, you'll see the serotype, and then you'll see an outbreak code that we have identified at the national level that's been downloaded in the local database, if there is one. And that's always good to know too, because maybe you have a new sequence in your database and you say, oh, well, it matches this ongoing outbreak investigation. I need to let my epidemiologist know. So these are just some screenshots of what people would see at the local level, like if they're using allele codes to do searches. So this one, we're wanting to see if it's a match to an ongoing cluster. So what they will do is they pull that isolate into a dendrogram, and you'll notice that 3 of these match to the 6th digit, and then 1 is to the 5th. So you want to still verify what the allele range is in a dendrogram, and here it's 0 to 1, which is extremely close. So we would notify our local epidemiologists about this match. And then this is another one. Let's say we're doing a cluster detection. You know, we look in the past 60 days and we find these 3 that appear to be close, at least by allele code, and we see that their range is 2 to 7 alleles. So we would report this new cluster to our epidemiologists. Another great, this is one of the biggest tools I think for allele codes, is being able to look at historical matches. So I can just query the database for something text. That way I don't have to tell the database, okay, I want to compare all these allele calls in this one isolate to everything. That takes a lot of computer power, it takes time. You know, all you got to do is have a network glitch and everything that you were searching for went away. So this is way easier. Not only that, but this information is shared within Cedric, that system I told you that the epidemiologists use. So you can actually just go there and do a quick text query and be able to look up everything that had this allele code in the past. Now, I do want to verify this in a dendrogram, and I look at these, and so I would let my epidemiologist know that there was a chicken isolate back from 2019 that matches this ongoing cluster. This could potentially lead them in the direction for finding the source of this current outbreak. And then this one just kind of shows, as you get, you know, you'll notice that not all of these allele codes are exactly the same. The top few share all up to the 6th digit. So that's kind of like, okay, these are 0 alleles, they're all indistinguishable. I know these are a cluster. But then I have this other one that only goes to the 5th digit, and then another one that doesn't even, it only goes to the 4th digit. But this is why we ask them to look at the 4th digit, because now you're including these and you're only going out to 8 alleles. So we would include all of these in our cluster search, or in our cluster report. All right, so we've gone over PulseNet and we've talked about allele codes, but I really want to talk about how this is used in cluster detection and outbreak investigations. So overall, we use annotated dendrograms and similarity matrices to investigate clusters. These are communicated back to the public health laboratory scientists as well as epidemiologists. And sometimes this shows that clusters are close, but genetically different enough that they could be investigated separately. And an example of that is below, where you have what looks to be, they're all very close, but they are in distinct groups. So CGMLST is the primary cluster detection method, and in general, what we, as a guideline, we ask for local clusters to be sequences within 10 alleles, and then at least 2 of those sequences should be within 5 alleles. And all of this, you still have to keep in mind, allele differences within a cluster may be larger or smaller, depending on the organism and the epidemiologic data. So this is switching a little bit. We are showing allele differences here, but we're also showing a resistance pattern. So here, the WGS analysis showed that a strain of Salmonella typhi was associated with travel to Iran and Iraq, and this was genetically distinct from a multi-drug resistant Salmonella typhi associated with travel to Pakistan. So the top clade you'll see has the travel to Iraq and Iran, and the bottom clade is travel to Pakistan. And we separated these by their allele differences. And another interesting point is that the different clades also show different resistance profiles, which is something interesting we've seen. This just shows where WGS was used in outbreak investigation of Salmonella Newport that ended up being associated with melons. The isolates with the earlier isolation dates formed one clade, and then as we got more and more, we noticed that another separate clade was kind of forming. Ultimately, because of the epidemiology, it was investigated all together, and it was the epi information was indicated a common supplier for the melons, and a traceback information from FDA indicated one single farm. But what this spurs is some interest in looking at enhanced surveillance for this particular melon growing region. So the WGS analysis will be used to compare strains from the region over time and how those might change. So switching gears to E. coli. So this is something that was a local cluster. So Arkansas did a cluster search in January, and they noted that this particular allele code had been seen 4 times in their local database in the past 60 days. This was unique to their database, and then they queried the national database to see if there were matches from other labs. And they confirmed that the allele range was 0, so indistinguishable. So it was definitely something they wanted to investigate. Turns out this was part of a larger national outbreak investigation. We gave it an allele code. It's part of a rep strain. I will go over rep strains in just a moment. And the allele range remains 0 alleles throughout the investigation. So that just gives you an example of a cluster with a single allele code. And I don't have any dendrogram pictures for this, but an outbreak was identified in 2019 that was linked to ready-to-eat chicken product. The WGS helped to link the cases and the ready-to-eat product. And what's very interesting about this, and so interesting about Listeria, is that it spanned over 3 years, which is something that we might not have been able to figure out with PFGE alone. Canada also had human matches, and they performed sampling and found Listeria in ready-to-eat chicken product. And then this is just a screenshot from the recall. So I just talked about a rep code, like what is that? So now I'm going to talk about what that is, what rep strains are, and how WGS is used for surveillance. So rep is an acronym. It just stands for reoccurring, emerging, and persisting. So reoccurring strain is just a strain that periodically causes a substantial number of illnesses, typically an outbreak separated by periods in which is not isolated from people, or it causes very few illnesses. So it might have this kind of bar graph up and down. Emerging is a strain that causes illnesses that have increased in frequency or have potential to increase in frequency over time. And persisting is a strain that causes illnesses consistently over time, something that we might consider like a very common strain that we're just always seeing. We see a lot of that with Enteritis. So for your information, PulseNet started assigning rep codes in December of 2019, and we've been tracking these in the PulseNet national databases, and these are also viewable to our public health partners via Cedric. So our PulseNet database managers assign these codes along with additional input from our reference lab as well as epidemiologists. And this just gives you an example of what a rep code looks like if you were to happen to see one. So it tells you immediately it's a rep code because it says rep at the beginning, and then it has the serotype code followed by a number. And so this rep EXH01 just indicates that it was the first coded rep of E. coli 157. And then it may or may not have a dash number, and that would just indicate a particular clade within that rep code. And this really just helps us identify and look over time, what we're seeing with our WGS data. And we've already talked about future studies that would include source attribution as well as looking at the evolution of the sequences over time. Just some current rep strains we have are Campylobacter jejuni. That was a strain from the MDR outbreak linked to puppies. 0157 linked to that Yuma romaine outbreak in 2018. Salmonella Kentucky, Salmonella Redding linked to various turkey products, and Vibrio parahemolyticus linked to reoccurring outbreaks linked to raw oysters. So what this is a minimum spanning tree, and these are really good for helping us visualize a lot of data. So this particular spanning tree includes over 850 isolates, and you'll see that there are 5 different subclades involved in this particular rep. This is the Yuma strain rep. And you'll notice, as you would expect, that each clade is a different, or each subclade is a different color. So you have your major one is in red, and then you have your other colors identifying the different clades. And this just shows that ultimately they're related, but they do span off. There's been approximately 20 different outbreaks associated with this rep strain, and sources have included beef, romaine lettuce, leafy greens, and as well as a lake exposure. And sequences will be included or excluded from these clusters based on relatedness to other uploads, but not just the allele code. And this is just a graph over time showing this particular rep strain. And as you can see, the largest number we had was during that 2018 Yuma romaine lettuce outbreak. I did want to note that we didn't fully move over to WGS as our subtapping method until July of 2019. So any past numbers like 2016 looks very scant. It's just because we didn't have the data. So in conclusion, PulseNet's a network that works collaboratively with food regulatory agencies and foodborne laboratory scientists and epidemiologists to detect and investigate foodborne disease outbreaks. WGS is used as a tool in detecting foodborne outbreaks as well as discovering reoccurring, emerging, and persisting strains, or those rep strains. Allele codes, outbreak codes, and rep codes are all tools used to track and identify strains and outbreaks in the PulseNet national databases. And thresholds for cluster identification are ever changing as we collect more data and learn more. So thank you for your time, and I'll take any questions you have.
Kelly, that was absolutely terrific. You did a tremendous job covering a large amount of information, and it's impressive to me to see how far you and your group have gone with the PulseNet in these rep strains, the being able to use virulence markers now and identify those through the whole genome sequencing, antibiotic resistant typing. It's incredible. So thank you for that. Let's see if we have any questions. I don't see any. How about with the antimicrobial resistance typing now that you're able to do directly, how is that working? Are you able to better focus now on highly virulent strains than you have in the past?
Yes, we had to have some updates to our software and our plugins late last year to really be able to visualize the antimicrobial resistance data within bionumerics. Now, and why that is important is just because before we would have to use an external system, and at least for our public health users, it's just easier if they have it all in one place. And so we finally have that, and we are working really closely with our NARMS group within our branch to look into this more. We honestly haven't done a whole lot. I mean, we have done some work with that typhy, but I know Jason Folster within NARMS, he has some alerts set up for any kind of XDR strains that pop up. But we are excited to see, what more data bring.
So where are you going next with this system? What's the next revolutionary step that you see?
Well, let's see. We are looking to reboot our software. What does that mean? We're at the very beginning stages of that planning, so I'm not even sure what that means. I know there's been talk about putting all the data in the cloud, making it more accessible. I know we do want to have some outward-facing abilities. Everyone wants more and more data, and we do provide data to NCBI, but there's not a lot of metadata associated with it, and so it does make it difficult for people that are outside the PulseNet network to be able to view this data in a real meaningful way. So, those are all kinds of things that are on the horizon. One other thing too is also metagenomics, being able to not have to rely on a culture as we move forward.
Terrific. Well, that's about as much as we can accommodate time-wise, but if you have, you in the audience have questions of Kelly, hopefully she'll be accommodating, and we'll share those questions with her and she can respond directly with you. Kelly, thank you again. And you mentioned the term metagenomics, which was perfect because that brings us into our next talk, which is going to be presented by Dr. Hank Denbacher, who is an assistant professor of bioinformatics at the University of Georgia Center for Food Safety. Hank has previously been at Cornell University and Texas Tech University, and he's authored many publications on genomics and population genetics of foodborne pathogens and spoilage organisms. And his current research now focuses on the novel field of food safety informatics. And Hank is going to share with us some of the recent advances in the area of metagenomic approaches to detect foodborne pathogens. So Hank?
Okay.
Hank,
you
can I
hear
get
me
you to hit
properly?
that swap displays
I
button real quick?
Yes, I will swap again. Excellent. Okay. So, yeah, that was a wonderful bridge. Let's see. So today I'm talking about advances in metagenomic approaches to detect foodborne pathogens, or metagenomic approaches to get to know more about the ecology of foodborne pathogens. And so this is the overview of my talk. I will have a short introduction or about metagenomic approaches, so different types of microbiome sequences. And while discussing those things, I will also stress the advances that have been made in microbiome sequencing approaches over the last couple of years. If we're talking about metagenomic approaches, we have to think of a trifecta. So it's a trifecta of the data we collect, sequencing technology, and how we analyze the data. So my next topic will be about the introduction of long-read sequencing, I think in the last couple of years in microbiome applications. And last but not least, I'm going to discuss some real-life data to show how we can leverage actually the microbiome for pathogen detection. So if we're talking about microbiome sequencing, there are two main ways of sequencing microbiomes. One of them is Amplicon sequencing. So that means that we use a highly conserved yet variable part of the genome, and about 300 to 500 base pair, dependent on the sequencing technology that we use, the sequence of all, or the preferred part of the organisms that we're interested in of a sample. And traditionally, those are rRNA genes. So 16S for archaea and bacteria, but one thing to keep in mind is that chloroplasts also have sequence 16S, and 18S and ITS for eukaryotes. So a quick overview. The wet lab part of the sequencing is basically doing a PCR and preparing the PCR in a library prep and sequencing it on your sequencing system of choice. A bioinformatics step where we do OTU classification. So this is a part of microbiome research where we have seen a lot of advances over the last couple of years, which consist of data filtering out cross contaminants, false index pairing, etc. And one of the things is, if you do this kind of sequencing, so Amplicon-based sequencing, is to put in a lot of controls, so negative controls and positive controls. So one of the advantages of Amplicon sequencing is that you can start with small amounts of starting material, which is, especially if you talk about environmental sampling, for instance, in food processing plants, very important. Based on the primers that you use, only the organisms of choice, so only bacteria, will be sequenced, and it's highly efficient. So because you only look at one gene, a limited number of reads is enough for a microbiome survey, which translates to, if you multiplex 96 samples on an Illumina MySeq run, to dependent on how what you pay for your consumables, you can push the price down to less than $40 a sample. The disadvantages of using Amplicon sequencing is that all the ways of most bioinformatics applications for Amplicon sequencing workflows rely on database searches. So you have to have a really good database with representatives of the organisms you expect in your database to classify your organisms. The reliance again on primers makes it also possible that you are missing organisms that are important for further analysis because your primers didn't catch that diversity. And one of the things that I found is a disadvantage of RNA amplicon sequence is that it also amplifies a case of 16S organelles of eukaryotes. So if you want to look for the microbiome of a living plant for endosymbionts, most of your reads will go to 16S of the chloroplast of a plant. So the other big way of sequencing microbiomes is shotgun microbiomics. So in that case, all DNA in the sample is sequenced without any enrichment or PCR amplification. So you treat the sample as if it was one large organism with a very big genome. Of course, the genome consists of multiple genomes of microorganisms. So there are two main approaches for analysis. You can do read classification. So that in that case, the bioinformatics workflow predicts per read, try to match it to what organism it belongs. And that's, again, very database dependent. And then there is the de novo assembly and binning approach. And that's a very interesting approach. And here we have a cartoon. Basically what it does is, here we have our original microbiomes, and these circles are individual genomes. We sequence them, and basically we don't sequence genomes in long reads. Usually we sequence them in little puzzle pieces of 300 base pairs, 250 base pairs if we use different sequencing technologies. We can have it up to like 10,000 base pairs, but we never sequence those genomes entirely. What we do then, we sequence them, we assemble those genomes and try to get reassemble those pieces of genomes in longer pieces, and then we try to classify those pieces based on abundance, so statistics, and on sequence characteristics. So in this case, this example is metabed, and we use tetranucleotides, so 4 base pair word frequencies to make individual bins, and then we make those bins and try to reconstruct the original genomes again. So this is a very interesting application. So what we get in the end from this workflow are MAGs, so metagenome assembled genomes. So the advantages of shotgun metagenomics is that we don't have any reliance on primers. Everything is sequenced, so we don't have to worry that our primers didn't catch like all of our diversity. And whole genomes are sequenced, so it makes it possible to study gene abundance, the presence of gene families of interest, so you can predict potentially virulence genes or antimicrobial resistance genes in your microbial population, and the ability to study the uncultured, the microbial dark matter. So it's better at that than amplicon-based approaches. And I always like to show this picture. This is like a tree of life, and all those red dots here, including this large new candidate phylum, are new organisms that have been discovered with that assembly-based approach. So the disadvantages is, again, it's almost like what's an advantage is also a disadvantage. Because we don't rely on primers, everything is sequenced. So if we go to a meat processing plant and we sequence our DNA. 99% of our DNA or of the sequence reads may go to actual animals that are processed in a plant. So in this case, chicken or turkey. And because we need a lot of, like, we don't look at a single gene, but at whole genomes, it's also expensive. So to get enough coverage for certain communities, especially complex communities, and an entire sequencing run is sometimes necessary. So instead of that $40 that we spend per sample for 16S sequencing, if you have a super complex microbiome, you may spend like $1,000 to $2,000 per sample. So one of the advantage that have so there are a lot of different variants of these two ways of sequencing microbiomes, getting a picture of what's in the microbiome. And one of the advances, I think, with respect to foodborne pathogens that has been pursued by the Deng Lab at the Center for Food Safety at UGA and by the FDA is the introduction of quasi-metagenomics. So one thing to keep in mind with foodborne pathogens, if things didn't get out of hand, is that they usually make up a very minor component of a microbiome. So if you want to do shotgun metagenomics, you need a lot of sequence capacity, even to find those foodborne pathogens. So they can easily go undetected in traditional microbiome analysis. So quasi-metagenomics uses a number of amplification and selective steps to sequence organisms of interest. So here's an overview, and this was a paper that was published in 2018 by Xiangyu Deng's group. In this case, they took 3 different products: black pepper, lettuce, and chicken breast. And what they did was first they put them through a traditional enrichment. In this case, they looked at Salmonella enterica. So Salmonella enterica enrichment. Then instead of going through the whole enrichment, they could stop the enrichment at a after a very short time, like 12 or 24 hours. And then they enriched the final sample with immunomagnetic beads that were specific for Enterobacteriaceae. And to top it off, they created more DNA after the enrichment with immunomagnetic beads with a whole genome amplification step. So with the 529 DNA polymerase, phage polymerase. So what you can do then, you can do real-time PCR to go straight for your organism of interest. You can also sequence your organism with your sequence platform of choice. And the interesting thing here is that they spiked the samples with known strains, and they were able to recover like the entire genome at an accurate enough level to do strain identification and do the kind of strain level subtyping that was discussed in the previous talk. So the other advantage, if we talk about sequencing, is the introduction of long-read sequencing technologies into metagenomics. So a lot of what we do as bioinformaticians, and I specifically say we because I consider myself a bioinformatician, a lot of uncertainty in microbiome datasets has to do with the fact that we use short reads. So Illumina reads are typically like between 150 and 300 base pairs these days. They're really accurate, but they're very short. So all our approaches we're dealing with small pieces of genome, and they just contain less taxonomic information. So we have to do a lot of bioinformatics tricks to name them. So replacing or including reads obtained with long-read technologies can greatly improve the accuracy of some of those microbiome analysis. Like I showed you the MAG workflow, so the assemblies are better, so we can better recover whole genomes from microbiomes. But we also have more data to make more accurate predictions of what is in our microbiomes. So the two different analysis workflows just get better. The only problem is that long-read technologies, as they are now, and again, that advances almost every day, is that they have a higher sequencing error rate as compared to, for example, Illumina reads. So here is an example of one of the early players of long-read technology. So Pacific Biosciences with their smart single molecule real-time sequencing. So it's a long-read platform, and one of the things to keep in mind is that it has a very large footprint. Even their smaller sequences have a large footprint, and you need a lot of specialized equipment for library preps. So kind of the new kid on the block, at least a couple of years ago, was Oxford Nanopore, and they produced these really small sequencing devices that you can plug into a laptop and do your sequencing in-house. And here we have the MinION, and here we have, I still haven't seen it in real life, the Smidgen that they developed as a sequencing device that you could plug into your iPhone. And this is an interesting long-read technology. So there are competitions of users to get the longest reads. So usually the long reads are between 100,000 base pairs. But recently I've heard people that could get long reads that were close to half a E. coli genome. And the cool thing about this data, this platform, is that it's fast. So you can do a sequencing run in a day, and even after a couple of hours, you can start analyzing the data. So while the sequencing run is in progress. So if you want to look for a specific organism, you can start to analyze your data while it comes off the machine. Problem is that it has a hyper read error rate. So as a bioinformatician, you have to deal with that. And the applications as they are now largely overlap with PacBio, but with a much lower cost in upfront investment. And just as a something to notice is that this is currently the sequencer of choice in a lot of SARS-CoV-2 sequencing efforts. So then I come to my last part of the talk. So we talked about looking for foodborne pathogens in metagenomic or microbiome data sets. We looked at how to amplify our pathogens of choice in a data set, but can we just use the microbiome data as they are without much manipulation to predict the occurrence of pathogens? Again, to keep in mind is that you don't find a lot of reads in your data sets of foodborne pathogens. If we have those microbiomes data, can we find higher abundance microbial taxa, families, genera, species that are accurate predictors of the occurrence of foodborne pathogens? So can we find novel indicator species with our microbiome data? Can we look at the whole community and associate like certain community profiles with occurrence of foodborne pathogens. And I'm not going into detail on that question, but that's certainly something we can do, and we can even do it with 16S data. So here I have an example of a group of Penn State, of the COFAG lab, and they looked at the microbiome of 3 apple and other 3 fruit packaging facilities to look for, and they used Amplicon-based methods, 16S for bacterial organisms and fungal ITS communities, to see if there was a relation to the presence of Listeria monocytogenes. And they did find that the microbiota in this facility, and specifically in the wet processing area, was associated with Listeria monocytogenes, and the highest Listeria monocytogenes occurrence was uniquely, those microbiomes were uniquely predominated by the bacterial family of the Pseudomonaceae and the fungal family of the Dipodaceae. So the interesting thing here is that there's this initial, and that's where bioinformatics comes in. Right now we have information at the family level, but we can use a novel taxonomy, so more precision in our taxonomic placement, and newer bioinformatics pipelines to go even deeper into those sequencing data to home in on potential species level associations, if possible, that can tell us more about those communities. So I used data too, so I reanalyzed their data set. And it's a method to infer the exact sequence of templates of the Amplicons in an Amplicon method. And it gives us Amplicon sequence variants. So we reconstruct the actual templates that gave us the PCR product. Next, I analyzed it with DSEC2, and that's a statistical method which tells us which of those sequence variants are significantly over or underrepresented in specific groups. So I compared samples that were Listeria monocytogenes positive versus those that are Listeria monocytogenes negative. And next, I looked for did a phylogenetic analysis of those partial 16S sequence to figure out what the diversity, how far we could go to figure out what the species or subpopulations were that contributed to those Listeria monocytogenes associated taxa. So here we have the output of DSEC analysis. It's a kind of a difficult picture, but all these dots are individual taxa that are significantly associated with the positive occurrence of Listeria or negative occurrence of Listeria monocytogenes. And so one of the interesting things here is that the genus that's mostly associated with Listeria here, positively, but also negatively associated with Listeria, is a subgroup of Pseudomonas E. And you can see here that apparently not all Pseudomonas E strains are created equal, because some of them are positively associated. So this is phylogenetic tree of Pseudomonas here, and some of them are negatively associated, and some of them are more abundant with when you compare them between environments with Listeria monocytogenes than others. So one of the things here is that is of note is the Pseudomonas fragi clade. So a cold-tolerant Pseudomonas that's very good in biofilm formation, which seems to be highly associated with Listeria monocytogenes. So those Pseudomonas fragi, if we look at could be like an indicator species that tells us more about harbored sites, potential harbored sites of Listeria monocytogenes, without having to search for Listeria monocytogenes. So this is just an example of how bioinformatics and improved bioinformatics methods that are improving over the years are contributing to our understanding of foodborne pathogens and their role in relation to the microbiome. So in summary, I hope I've shown you that microbiome studies can help us to advance detection of foodborne pathogens. At least they can help to advance our understanding of the ecology of foodborne pathogens. And I think the main advantages in microbiome research are made in sequencing approaches, advances in sequencing biotechnologies, and advances in bioinformatics. And what I want to mention last is that the public availability of those data sets, even old data sets in repositories like NCBI's SRA, make it possible to go back to some of those old data sets and retroactively apply some of those advantages, especially the advantages in bioinformatics, to old data sets. And I want to thank you for your attention. That's it.
Hank, that was a terrific overview and bringing us up to date on where metagenomics stands. If you have questions, please let me know, but I'll start them out. We have several different approaches that can be taken in metagenomics and detecting pathogens in clinical specimens, but also in foods. And first of all, how sensitive can these be? We're talking about culturally, we want to detect at least one pathogen in 25 grams of food, or even more sensitive. And secondly, cost. And thirdly, the practicality in doing routine testing in the microbiology lab for food samples.
Yes, and that's I mean, all those things are very good arguments. I think we're not there yet. Like right now, I think it's mainly academic. And for instance, what I didn't mention is for a lot of food matrices and for a lot of foodborne pathogens, like the DNA extraction methods really make a big difference. So in my lab, and I think in a lot of other labs, one of the reasons probably that you don't find a lot of reads of your foodborne pathogens is that a bacterium like Listeria monocytogenes with like a generic DNA extraction protocol doesn't lyse as quickly as most of the other members in a microbiome. So just as it's an artifact of DNA isolation. Right now, I know that there are commercial packages that help you with the analysis of microbiome data sets, but it's not all standardized. It's not there yet, in my opinion. It's interesting to do, I think, sequencing your processing facilities, products, all those things can help you to understand the microbiome of your product and to detect events probably that lead to or associated with the introduction of spoilage organisms or foodborne pathogens in your plant. But I see that all as currently as individual cases, not as like standard detection protocols that can be applied to each and every specimen.
Gotcha.
Yeah.
Well, we're about out of time, unfortunately, Hank. This is a really interesting and important topic for the future of detecting foodborne pathogens. it's not going to go away. We're going to, I know, see more and more advancement in this area. So thank you for sharing that with us. And again, to the audience, if you have questions, please submit them, and we'll pass them on to Hank, and he hopefully will get back with you. So this brings us to our next presentation, which is going to be given by Dr. Mark Allard. And Dr. Allard, he's a Harvard graduate, and he started his career in teaching at Washington University, where he was there for 14 years, and then moved over to the FDA, and that was in 2008. And I credit him in large part to be the foundation and brainchild behind the genome tracker, which he's going to share with us, that was developed at FDA and enables source tracking of these pathogens in foodborne outbreaks. So with no further ado, Dr. Allard, I look forward to hearing from you regarding the advances that you have made in not only the genome tracker, which is largely used in the US, but also the Galaxy tracker, which, as my understanding, is used internationally as well. So we can begin to better source track pathogens that are involved in foodborne outbreaks. With that, thank you. Thank you for joining us, Dr. Allard, and please enlighten us in your new findings.
Thank you very much. And so everything I'm going to be talking to you today about is using genomics to provide safe, wholesome, and sanitary foods. You heard about Kelly talked about the CDC and PulseNet, and essentially what I want to talk about is the landscape where FDA and USDA also share all of our pathogens, all of our genomic data. And essentially, and then we also uploaded to the SRA at NCBI. And so essentially, one of the first lessons we learned is you need to share your data in a timely way. And the sooner you share it and the more metadata, the bigger impact you'll have on reducing public health problems. And essentially, a One Health approach, genomics can be compared across, regardless of host, they can be directly shared. And so it integrates with this One Health method. Where's my slide here? So once the data is at NCBI, it can also be used by many other software tools. And so, because NCBI can only do so much, they're sort of thinking about it in a global way, and they're also storing and sharing the data and so essentially curating it for the long term. And these other groups can all add value to their particular stakeholders and their particular questions. And so, and this includes, global databases. Some of these are wide open, and others you have to be a member to participate. But this way we get more value for the data that we upload. So in particular, the FDA wants to make sure that all the data can be optimized for use across many platforms. And this idea is talked about as the FAIR principles, which means findable, accessible, interoperable, and usable. And so this is sort of a constant work in progress of keeping standards up, and as new platforms and new data files come out in the research industry, is to integrate those so that the data can be shared across multiple platforms. And you mentioned Galaxy Tracker. What FDA has done is we've used an open platform. Galaxy is an open platform, a cloud-based computing, and the Galaxy Tracker is just software that FDA uses that are essentially tools, custom workflows, and these are all the things that the Genome Tracker members do. They check the quality of the data, they can upload, they can do many of the basic pipelines, and this allows anyone in the world to fully see in a transparent way exactly what our data analysis pipelines are doing. And so not only is all the genomic data publicly available at NCBI, but all the bioinformatic methods are transparently shared. And then lastly, we've been starting to use, this is work by Ruth Timmy and Maria Balke, the Protocols.io is a place where we can share all our literature and share all of the protocols, all of our standard operating procedures. So essentially, anyone with a sequencer anywhere in the world can essentially plug in and use these tools, get access to how we do it, and then they can also either be a part of our network or they can do it independently. You know, they may want to do work that they don't want to share. But this covers all kinds of levels. The protocols include how you do the quality checks, how you do the metadata, which is the description of the isolate that you're sequencing, and then detailed on data submission and data curation. And this way, we have standards, and so fewer people make mistakes, and you receive data in a sort of organized way. Essentially, the lessons learned is that when you do these standardized methods, you see fewer problems, and essentially open tools, particularly open software tools, promote global adoption because generally they're less expensive than buying a piece of software, maintaining your own computational costs. For all of our partners, we just pay the computational fees for running the Galaxy Tracker. So in this case, there's no cost to our collaborators. By sequencing and uploading their data and participating in the Genome Tracker, we found that this is a good way to essentially provide more bioinformatic tools to our partners. There are other cloud-based options. This also, you know, you could give a copy of all the software to give to another group, another food safety agency, you know, and they can either run it themselves and pay those computational costs, or they can be part of our network depending on what we work out. It also controls the data analysis for version control, because a lot of this software has a lot of choices. And so this is a way that we can define and verify a platform as doing what we think it can do, and then share it so that different groups aren't using different versions of the software. And so this is mostly genomic, epidemiological, and analysis tools that we share, as well as quality. The total number of sequences in the Genome Tracker database has continued to rise exponentially. We're nearly, I think we just broke 600,000 foodborne pathogens. There's another 200,000 of other pathogens. And this, I want to reaffirm, this isn't just FDA, this is all of their partners, the CDC, the FDA, the USDA, but it also includes all of our international and domestic partners, as well as anyone who essentially publishes a salmonella genome and puts it in GenBank. That goes into the database. So we're upwards to 12,000 isolates a month or more, and we continue to expand essentially the diversity of pathogens and the total numbers of pathogens. I will say we primarily focused on bacterial foodborne pathogens, but one of the future goals is to start also collecting more virus and parasite, foodborne virus and parasite data. But we're just at the beginning stages of that. So at NCBI, essentially, the first application is that the phylogeny identifies novel linkages for outbreak detection and infectious disease control. And so if you upload genomes to NCBI, they produce a daily cluster of the very tips of the tree. So this is only the very tips of the tree within 50 SNPs or less. But this is the application that FDA and their public health partners are most interested in, and that is here's an example where an FDA inspector swabbed and found 2 positives at a mung bean sprout facility, and we uploaded that, and the cluster shows that it's clustered with some clinical illnesses from PulseNet. All these PNUSA are the PulseNet USA clinical samples. And so, you know, there's really 3 things we're looking for. The first is the new inspection data positives. Did they link to anything, any other food or any clinical? That may be the first tier. The second would be, has it made anybody sick in the past? Or, I mean, once again, this is a hypothesis that the contaminated food may be getting and making people sick. But even an old isolate suggests that there's a risk that pathogen has caused disease in the past and may in the present. And then the third sort of tier of concern is whether there are current recent clinical cases linked. And so then that means that we need to really dig in and look carefully at this cluster. And at NCBI, there's over 36,000 clusters examined daily. About 4,000, maybe almost 5,000 of the clusters has an FDA isolate sitting in the cluster. So the cluster is the first piece, and this is what genomics provides. Fewer clinical cases are needed to have confidence that they're interlinked. This early and ambiguous determination of the scope of the cluster, you know, the clusters that don't group in that didn't share the same common exposure to a food, and any unrelated clinical isolates evidence that there's an independent co-occurring contamination. So it's either completely independent or there's polyclonality, meaning multiple lineages of pathogens that come from the same contamination event. And so, as spoken by, a lot of this will overlap with Kelly's talk earlier, it essentially allows previously indistinguishable serotypes and PFGE types, it allows it now to be tracked and genetically linked. And essentially, this higher confidence incentivizes early actions. So, and I mean early actions both by the food producing firm as well as the public health agencies. And essentially the genomic evidence, what I would call the microbiological laboratory piece of evidence, the clustering, is just a part of the evidence that's typically used to identify an outbreak vehicle. There's also all of the epidemiological evidence, which is once they define the scope of the outbreak, they can determine the association between the illness and the food exposure. And then the third piece of evidence is the additional traceback and investigators. This is food inspectors going back to facilities and essentially trying to determine the common source and more importantly, determining the root cause of the illnesses, how the pathogen is getting into the food supply. So this may lead them, depending on where the first positives occurred, or they may go back to a processing plant, and that may be lead them back to a farm or even specific ingredients. So the FDA inspectors are using the genomic data and the epi and their own inspection to look for the pathogen. And this is what we call trace forward and trace backward, because the FDA wants to know essentially if there's a contaminated ingredient, it may have been shipped to multiple food production facilities. And so you'd want to check which of the facilities, some of those facilities may have received contaminated ingredient, but they controlled it, whereas other food production sites or places, somehow it got past their preventative controls. And so we want to know not only where did all the potentially contaminated food get shipped to, but also going back as well. And so this is a detailed part, and essentially for any outbreak, they would want to go do this kind of study in a post-outbreak response to understand what happened and how the pathogen entered into the food supply. And so essentially the source tracking and root cause event investigation does a very important thing. It identifies whether this is a resident or a transient pathogen. So we know that the fields are not a sterile place. There's animals going through here, there's water and soil, there's pathogens on the farm. You can't keep that from happening, really. It's difficult. But what can happen is that there could be a farming practice that essentially causes a man-made occurrence of creating more pathogens or that you can control it. And this is often directly related to animal waste, fertilizer, soil amendments, and the treatments of water. Within the facility itself, if you get one of those pathogens that's in from the field get lodged as a resident pathogen in the facility, then you have a serious problem, because if it's in equipment or then it can regularly sort of spill out and get onto the food product, and then the company's actually producing more contaminated food than because of this activity. So determining the resident and transient stature of the pathogen is important, and then figuring out a preventative control to interrupt it either on the farm or in the food production. And essentially, the increased identification of harborage and root cause of contamination is directly supported by genomics. And the FDA notifies the responsible food facility essentially that they think there's a pathogen and that they're the cause of the problem. And this increases awareness, and awareness incentivizes the food manufacturers to invest in preventative controls and essentially keep investing until they have preventative controls at work. And so the new root cause discoveries also improve his knowledge of reservoirs and risks. So not just to that food industry, but the FDA will publish this in their good farming practices and good food manufacturing practices. So it doesn't just affect the one particular firm that had a problem, but it notifies the whole food class. All the almond growers or all the leafy green folks will hopefully learn from any one incident. And then the FDA Office of Compliance, they're watching this, just like Kelly talked about the rep strains. We're watching known strains from past events, and essentially we're watching that. And if that preventative control somehow fails again, or it's not really working 100%, then we'll see those pathogens show up in the clinical community or in food, and we can rapidly trace back to its original source. And essentially the combination of zero tolerance for many of these pathogens and the power of whole genome prediction is reducing foodborne illness in the US. So these tools are working. some of the time you can't figure out what the root cause is, and from just an initial inspection. And so some of the time the FDA gets involved with research, which are essentially longitudinal studies. And this would be, in this case, this is a study that went from 2010 to 2014 in the Delmarva Peninsula, where they collected many samples essentially on the farm, looking for where the pathogens reside. Where can you find pathogens? What are the risks? And in this particular study, all the green mean they found no salmonella, but the red were places where we regularly found salmonella. And this was in the creeks and sediments. And most standing fresh water and some of the animals that are like regularly in the water, like the waterfowl, you could find the pathogens. And then this got us a very careful study of, well, if the risk is in the standing fresh water, then how are the farms using water, and what are the preventative controls in place to prevent foodborne pathogens in the water from getting into the crops? And so this helps with guidance, but it's typically a long-term surveillance and research. And we also work with food communities like the Leafy Green Task Force and their academic collaborators. So all of our data, publicly facing, all of our data is at NCBI, and we talked about it. This is, you don't need a login or account. You can go and see any of this information. This is our publicly available data. And I wanted to point to the right side of the slide. There's lots of information here about antimicrobial resistance monitoring. They have whole databases of the 5,000 genes known to cause antimicrobial resistance. And you can actually get a daily check of what new genomes have come out with new antimicrobial resistance monitoring genes. So all the clusters are here as well. But this is a set of tools that I encourage you to go and look at. And one of the things we've been working on carefully over the last few years, but in particular, is the metadata. So one of the important things to do is essentially fully describe your metadata in a standard way. And so we have various isolate attributes, but it includes the collection dates, the species, essentially who collected it, some geographic area. We typically list it down to state, and then the source is a critical piece. We break everything either as a human clinical or a food or environment, and this is a way where we can then have automatic software that says, when do we see a match between a clinical and a food or an environmental sample? And then more detailed information about the labs and the exactly what kind of environment it was taken from. And these are all deposited through the NCBI system of bioprojects, and this is what's called a biosample. So the more we can organize the metadata and standardize it, the more powerful it can be used for statistics. And so this is Genome Graph R, is a risk prediction software that was created with collaboration between France and the US FDA and some academics. And this uses this ontology where you say, is it food or is it a plant? Is it environment? If it's a food, is it a plant or an animal? And it goes all the way down to, a particular kind of fish or bivalve. That you can, and there are different kinds of ontology, but essentially this makes all of that data more valuable for risk assessment and any kind of statistical analysis. So essentially, recently we went back, we didn't do this originally, but we've been back to all 90,000 FDA isolates in the NCBI, and we added the food ontology and the IFSAC ontology. But there are other ontologies as well, the GenEPO. It's exactly where in the environment do you collect, and how you name the food. And this allows the FDA to say and make predictions, is there more risk from tree nuts or is there more risk from leafy greens? And those results may help us decide what to do. And I just want to say that it's not just antimicrobial resistance. In the AMR Finder Plus data set, there's also lots of genes related to other genes relating to stress and virulence, such as the persistence of a biofilm or the resistance to a particular kind of disinfectant, quat resistance, chlorine resistance. These genes can also be called for every genome uploaded into the database. So this is another set of tools that we hope to help the food industry to understand their resident pathogens and to then also look carefully at preventative controls to essentially control this natural evolution, or well, some of it's selected. It's living in these environments that some of the bacteria become resistant. Another thing we can do, and this is future vision, is, you know, we know how to do genomics, we know how to do source tracking and root cause investigation, and we can essentially educate a broader community on the power of these genomic tools. And so we have a big program the next 3 years in building food safety and capacity through the Asia-Pacific Economic Cooperation. And this is a long-standing network, global networks that we can leverage to essentially adopt genomics and to get them to all be integrated in this great food safety. So here's 21 economic, some of these are countries and others are collaborators, but across the Pacific Rim, and we'll be doing a lot of meetings like this to provide awareness, but then it'll get down to surveillance and actual laboratory training and bioinformatic training. The last thing I really want to talk about is, you know, the question is, why should you pay for this expensive genomic technology? And so we did an economic evaluation of whole genome sequencing programs in the US. And essentially we're comparing two data sets. We have all of the data at NCBI.
Mark, we've lost your voice.
Has been watching. So it's two data sets, NCBI and the clusters and the metadata, and then the other data set is the NCBI's tracking of all the outbreaks and the food associated and the number of people. And what this shows is that as we increase sequencing, essentially we see fewer illnesses. We see a reduction in the burden of illness. And this occurred a little slowly in the beginning. It takes 1 or 2 years for your investment to pay off, but by 2019, we saw a reduction in the burden of illness of almost $500 million. And so, and I suspect today it's probably much closer to $1 billion. And the pathogens, you can see the Listeria is showing the greatest amount of reduction in the burden of illness. And essentially what this shows is that whole genome sequencing is definitely an important link to food safety, and the most heavily sequenced pathogens, those illnesses are decreasing faster that related to other pathogens. Essentially the heavily sequenced pathogens are getting smaller, the outbreaks are getting smaller, though we're seeing more of them. And we think this is related to faster and more precise outbreak investigation with all of our collaborators within the US. And even considering the additional cost, the program likely paid for itself. So in addition to the direct effects on public health, to program increased accountability, it increases effectiveness and efficiency of our compliance and enforcement, and it facilitates root cause analysis, essentially risk assessment and risk management. and we believe that this would work in any other country, even though their own food safety culture and programs may be a little different. And like I said, we're not covering all of the foodborne pathogens. We're only sequencing sort of the top ones, but there's even more impact that we could potentially do. And then lastly, you know, what we're showing this evidence, we're saying if the size of the circle represents the amount of funding, you know, if you want more maximum benefit, then you need to do more funding in the food and environmental sampling and surveillance. And so essentially, as all the speakers have shown, WGS is reproducible, accurate. It's increases in surveillance should uncover more reservoirs, and essentially we're incrementally improving our food safety, and this will compound to fewer contaminations that reoccur. Every root cause we solve and institute a preventative control. And then there's all these other tools that Hank was talking about, emerging technologies like long-read portable, MinION technology, metagenomics. These are all mobile devices. These are all possible things. We know how to transfer this information, and in fact, WGS should be expanded to other human, animal, and plant pathogens. Essentially, it can also be translated and transferable to other infectious disease control applications like hospitals, nursing homes, medical manufacturing, waste management, composting, agricultural use and reuse. And many of the genome tracker labs pivoted and used their equipment and data sharing and uploading to directly to address pandemic response. And in fact, the FDA is going to also be sequencing for COVID-19 from sewage waters connected to and to protect food safety workers around the US. Anyway, thank you. This is a huge endeavor with a lot of people. This is just an acknowledgement for the water surveillance, and then there are lots of whole agencies where we're directly involved and collaborating. Thank you.
Thank you, Mark. That is really impressive what you have done. You and the other groups have done to use genomic science to basically help food processors do a safer job in producing food and overall reducing foodborne illness. It's incredible to me the advances that have been made, largely led by FDA, over this relatively short period of time. So congratulations and thank you for that. We have time for one quick question, which I'll make because I don't see any others. You've indicated that you're able to go back and identify a root cause. Now, with outbreaks, my impression is that we still have a ways to go in identifying root cause of outbreaks. Where do we stand in terms of making improvements in root cause in identifying sources of outbreak pathogens?
Yes, and as you know, just read the news. We're still having outbreaks that end. All the people got sick, and no specific cause was identified. And so that shows that you can't always do it just with epi. And so I think we need more environmental sampling. Essentially, we need to do more, invest more surveillance, because this works best when you have a repeat problem. And so, but if there's nothing in the database that essentially links back, then you don't really have additional evidence to consider. Even if we had a little bit of information, like we could say, we think this product came from Indonesia, it may help to identify and help the EPI questionnaires sort of focus their methodology. So without more sequence data and evidence in these databases, we're still going to just miss some of this, and anything new we might miss as well. So this isn't a total solution. We still have work to do.
Terrific. Thank you for that, Mark. in the interest of time, we need to move on to our fourth speaker, which is a topic that is at the forefront of everybody's mind, and that's the COVID, the coronavirus that causes COVID. And Leanne Jacobs, who is at North Carolina State University, she's a distinguished professor there, is considered to be one of the leaders in not just the country but in the world in the area of foodborne viruses, in particular norovirus. But she also has shifted some of her attention to the coronavirus, which causes COVID-19. And one of the big problems we've had in the food industry regarding this situation is that people in the workforce are becoming ill, and there's not enough people then to provide all the ingredients that we need to make food. So Leanne has addressed this, and she's going to share with us the impacts and risk of COVID-19 in the workforce of the food supply chain. Leanne?
Hi, can everybody hear me?
Yeah, I can.
Okay, awesome, great. I'm delighted to be here to talk about impacts and risk associated with COVID-19 in the food supply. I will just sort of start with my my obligatory conflicts of interest statement, which tells you what I have been involved in relative to potential conflict of interest, which really is not much. Before I launch into this, I really need to acknowledge Dr. Juan Leon, who is a faculty member at Emory University School of Public Health. He and I have together been working on putting the risk model together that I'm going to talk about at the end of this presentation. And particularly, Julia, his PhD student, has done most of the kind of boots on the ground work relative to this. So this is definitely a joint effort. But with that, I am going to start with the official statement about foodborne transmission. And Mike actually already gave this at the beginning of the symposium. I'll probably use the WHO statement, which is there is currently no evidence that people can catch COVID-19 from food or food packaging. And we stand by that statement, and that is very uniform throughout all of the agencies in the world. So this is not a foodborne pathogen. With that said, it has had significant impact on the food industry. And and that's been largely in the essential workforce. And so that's pretty much food workers. We'll talk a little bit more about that in a few slides. The first we saw of this in the US probably really was in the restaurant industry. But we also began seeing clusters of illness in meat processing facilities, and that actually expanded by probably the summer of 2020. We began seeing clusters of illness in other food manufacturing facilities, such as frozen fruit, such as seafood, even some prepared food manufacturing facilities. So clearly, COVID-19 was spreading amongst workers in these facilities. And there's a great study that was done in California that was able to kind of get a handle on how much this particular sector was impacted by the pandemic. And in the United States, we have this classification of essential workforce or essential workers. Those are kind of individuals that are necessary for health purposes, but also secondarily for keeping our world and economy going. And the food and agriculture sector is part of this essential workforce. And you can see here from this slide that they experienced excess mortality. They certainly experienced excess morbidity as well. And relative to some of the other members of the essential workforce, the food and egg sector was actually even more highly affected. Now, there are lots and lots and lots of things we can say about SARS-CoV-2, which make it kind of this unique pathogen. I am not going to go into a lot of detail on many of those, but I do want to make the point that, and it's a really important point, that there is a high level of virus shedding, both pre-symptomatically, so before people develop symptoms, and also in asymptomatic individuals. And that presents a huge issue because people are getting exposed to the virus from individuals that have no idea that they're infected. And that is an important, it's not certainly not the only driver of the pandemic, but that's been an important driver of the pandemic. So let's talk a little bit about transmission of SARS-CoV-2. This is a respiratory, largely a respiratory transmitted virus. There's some fascinating aerosol physics, engineering information that's come out of working with SARS-CoV-2. But basically what happens is certainly in the phase of disease where people are shedding a lot, most of the virus tends to be in the upper respiratory tract. And any activities that involve that, from as simple as breathing to talking, coughing, sneezing, yelling, etc., produce this turbulent, this multi-phase turbulent gas cloud, okay, which you can see on the right-hand side. And in that cloud is a combination of air and droplets of various sizes, and those droplets are from very tiny to very large. And the virus tends to associate with those droplets. So if you become, if you are exposed to the turbulent gas cloud immediately, you can directly contract the virus from exposure to the cloud. And then there are also indirect transmission routes. And this slide is a great illustration of what those indirect, or kind of the cadre of exposure routes to SARS-CoV-2. So here is an infected individual. They, let's say, sneeze, and they produce droplets of all sorts. And if a susceptible person is in close contact with that gas cloud, they'll directly get infected by inhalation. The very large particles tend to settle quite rapidly onto surfaces. They just drop. And that is the source of virus contamination for what we call fomite contamination or surface contamination. And that results in infection if somebody contacts the surface and then manages to contact their nose or their mouth, or they basically self-inoculate. The smaller particles in that plume will stay in the air for long periods of time, and that could be hours, sometimes even days. And of course, they're going to get diluted out in the process, but the bottom line is that if you have spent a significant amount of time in an enclosed area where you have these small particles floating around with virus association, it's also possible to become inoculated or become infected in that manner. Certainly, straight person-to-person transmission is also is possible, although that is not what we've done in our model, addressed in our model. So what has the food industry done in order to address this issue? Well, they've pretty much done most of the controls that we have as a community at large put into place. They've instituted these controls extraordinarily quickly. Testing was really done mostly in instances of clusters. Some companies are also doing a little bit of screening testing. Vaccination approaches are just being just being started. And you can see on the right-hand side of this slide that there have been just a cadre of physical distancing controls that have been put into place. Now, I mentioned that these were put into place, and in an ideal world, we would have had data to inform what are the best mitigations to put into place, where, when, why, how. We didn't. We had to act very quickly. And so you can see here on the left-hand side of this slide, these are just some basic practices, but there's a ton of uncertainty about what really is the most effective mitigation strategy to address those. And so, of course, as a risk assessment person, it makes sense to inform this kind of decision making with quantitative microbial risk assessment. And I suspect almost everyone in the audience here is pretty familiar with risk assessment, so I don't really need to explain this slide. But suffice it to say is that we haven't really been able to use risk models because we didn't have sufficient data to make the initial decisions that we needed to make to manage COVID-19. Now we have more data and we're starting to see risk models be produced. And I'm going to spend the rest of the presentation talking about a few of those. So this is actually a very early one. And I love this study. It's illustrative of the complexity of managing COVID-19, and it's also illustrative of a really simple but effective approach. So what these investigators were interested in was a cadre of potential controls, so low occupancy and high occupancy. They were also interested in ventilation, face coverings, or the effectiveness of face coverings. And they looked at, and also time period, contact time period. And they looked at how those all interface together to effectively express risk. And this is what's called a qualitative risk assessment. So there's high, medium, low risk. Red is high, yellow is medium, green is lower risk. And a great illustration of this would be for low occupancy conditions with good indoor ventilation. If people wear face coverings, they fall into the low risk category. But if they wear no face coverings, they fall into a moderate risk category, and that would be associated with a speaking action, for example. And you can make these comparisons across the board using this particular risk model. This is a very recent publication that was very interested in the relative importance of actions like breathing, speaking, singing, coughing, and sneezing. Remember, the size of the plume and how far it travels is all dependent upon the type of physical activity. And these particular investigators were interested in looking at the impact of ventilation or air exchange. And I'll just show you, for example, in the case of a sneeze event, they found that at an inoculum dose of about 10 to the 9th viruses, in a 2-hour period with no ventilation, the risk of infection was quite high, almost 100%. And when they were able to institute 6 air exchanges per hour, which is pretty moderate degree of air exchange, that risk falls down to about 50%. So relatively speaking, this shows how we were able to modify risk based on an intervention. So that leads me to the work that Juan and Julia and I and the Emory team have done. And we were interested specifically in a enclosed food manufacturing facility and how to manage COVID transmission within such a facility with respect to the essential workforce. We used as a model a frozen food facility with approximately 100 workers in the production line, and we asked two major questions: which transmission pathway was most important? Was it aerosols, in other words, the tiny droplets or tiny particles that last in the air for long periods of time, droplets, which are the larger droplets, or surfaces? And are the current worker protection strategies enough? And these were the worker protection strategies that we focused on in this risk modeling exercise. So to show you a very quick overview of the model, we began with an infected SARS-CoV-2 worker. We were looking at exposure of that worker, and then finally infection risk to that worker. We looked at aerosols, both large and small particle aerosols, as well as environmental or fomite sources of infection. And there were a number of factors that we investigated that were really important and for inclusion in the model. One is droplet size, because that impacts both distance traveled as well as how quickly something falls to another surface. We were very interested in distance between the infected worker and the susceptible worker. We modeled this within a 1 to 8 hour exposure that was indicative of a working shift. And as I said before, we looked at masking, we looked at air exchange rates, we looked at gloving and hand washing, and we looked at surface disinfection. Now, this slide just shows you sort of an overview of some, not all, of the parameters that were included in the model. A large number of the parameters we were able to get from published literature or literature, good literature that was in review. There were instances where we had to consult experts, particularly in the aerosolization area. And American Frozen Foods Institute was gracious and helped us get processing and facility dimensions, things like that, from their stakeholders. So I'm going to go back to our first risk modeling question: was which of these 3 transmission routes or pathways is the most important? And this is some of the early simulations that we did, and we were basically looking at total infectious SARS-CoV-2 on the y-axis as a function of droplet transmission, fomite transmission, or aerosol transmission, and distance. And overwhelmingly, we had the highest potential dose exposure at a 3-foot distance with droplets. And in point of fact, any of the other combinations gave exposure to a much lower dose of virus. So based on that, we were able to say that that droplet transmission appeared to be driving the outbreak in this particular venue, and that distance was a very, very important consideration. So with that, we went on and did some more simulations and looked at what the relative distance, what the relative difference in distance had based on, in this case, worker risk rather than number of viruses. And this is in association with coughing events. And if you note, at a 3-foot distance between worker and infected worker and susceptible individual, you can see here that risk, that risk increases with pretty substantially with time, but it is relatively low when you're at these longer or greater distances, and that is more indicative, of course, of aerosol exposure. And interestingly, risk hits almost unity. It's almost 100% if this distance of 3 foot over an 8-hour period of time is kept. We then looked at the impact of masking. We first investigated cloth masks versus surgical masks, and found them to be pretty at a distance of 3 foot between infected and susceptible individual. They provided risk reduction between 50 and 60%. If you double masked, you got about a 90% risk reduction, and if you used an N95, which is not used by this particular population, you could get it down to 99.9%. Okay, so that's at your 3 foot distance. As you increase distance, you get much better control, such that even masking with cloth or surgical reduces your risk to less than 1%. So masking is extraordinarily helpful. We also were interested in air exchanges per hour and its impact on risk with an 8-hour exposure. The industry standard is 2 to 6 air exchanges per hour in most of these facilities. We found, again, if at the 3 foot distance, air exchanges, as you increase the air exchange rate up to a high of potentially 8, you reduced risk in that short distance by about 50%. And if you were looking at further distances, you pretty much air exchange helped even pretty dramatically by certainly 4 to 6 air exchanges. And in point of fact, the contribution to risk reduction at the greater distance was not as overwhelming when as compared to the 3 foot distance. Okay, so once you've controlled or sought to control droplet transmission, then we wanted to also look at surface transmission, both associated with hand hygiene and disinfection. You can, a couple take-home messages here relative to risk. The contribution of hand hygiene and surfaces is much lower than the contribution of respiratory, maximum of a little bit over 20%. Although there is substantial reduction in that 20% risk by instituting hand washing and gloving policies, and also certain reduction in surface in the potential of surfaces to serve as a source of infection, particularly if you went down to every other hour of surface disinfection. So in point of fact, how do we actually do this? Well, in real life, we use multiple strategies. So the final simulation that we did looked at hand washing twice per shift plus sanitizing once per shift. And we combined that with distance, as well as with masking. And again, at the 3 foot distance, this kind of bundled strategy gave us about a 75 to 85% risk reduction. Double masking brought that up to 95%. And increasing distance, of course, brings the risk down to literally less than 1%. So bundled strategies work. So what's our summary here? Well, the focus needs to be, at least according to this particular risk modeling exercise, on droplet transmission at a distance of less than 3 feet. Once droplet transmission is addressed, surface transmission strategies to reduce surface transmission are effective. Combinations work. And based on our model, we really felt that the food industry practices do protect workers from SARS-CoV-2 infection. And I think the epidemiology is very clear that once these strategies were put in place, the clusters of diseases in these particular facilities fell pretty dramatically. So what are we going to do with this model going forward? Well, it's designed such that it can be used for a variety of other venues. We could use it in a restaurant setting. We can use it in other types of food plants with modification. Right now, there's a lot of interest in industry about the potential for transmission of SARS-CoV-2 in frozen food packaging. This is actually a big issue on the international scale. And so we are in the process of adapting the model to understand or to get a better feel of what exactly is the risk of transmission of this virus through frozen food packaging. And to just sort of wet your whistle for where we go in COVID management phase 2 as vaccination strategies are introduced in the population. We certainly want to look at the impact of vaccination and vaccination compliance on worker health in this particular workforce, and this model can definitely do that. We are also really interested in understanding and how, once vaccination becomes more widespread, what, if any, strategies can be relaxed, because this is what's being done now is really at great cost to the food industry as well as its workers. As we approach herd immunity, which is probably 85-ish%, how are we going to approach disease surveillance? You know, a little lower than herd immunity, we're going to still be seeing clusters, but we're going to have to be able to figure out how to detect those clusters, and that's going to be tricky. And that there may be a place for testing strategies relative to that. Certainly, we know very little about the variants and what their impact is going to be, and it's pretty clear that we're going to have to boost after initial vaccination, and so managing that is also a big black box. And with that, I will finish my presentation and be happy to take any questions.
Leanne, that was absolutely terrific, very timely and very useful information. So thank you for that. I think we have time for maybe 2 really quick questions. I'll start it out. Can you comment on the CDC's announcement of the chance of getting COVID infections from food surfaces being less than, I think it's 1 in 10,000?
Yeah, I haven't seen that particular announcement, but you know, it's interesting that you asked this question, Mike, because our motivation for doing the very first part of that risk exercise was really came from work I've done with neurotransmission from surfaces, and it happens, but it's not a very common event, and we had this feeling that it wasn't a really common event, but we needed backing for that. And I think that this model provided the backing that surfaces, while they can be a source of contamination, are not a certainly not driving the pandemic, and they're not a significant source. And I will tell you that we'll have a lot more information on that when we look at the frozen food packaging model that we're in the process of building.
Yeah, that's very helpful. Thank you. My concern is less than 1 in 10,000 sounds like it could be a lot. I don't believe it is, but that seems to me to be a pretty high likelihood. But you'll find out for us, so yeah, thank you.
I'll find out. You know, I don't think of 1 in 10,000 as being very high when your respiratory exposures are potentially, you know, 1 in 5. But you know, that whole issue of what's acceptable risk, you know, that's not a quantitative argument. That's a qualitative argument that has to be done by public health officials.
Okay, we're running out of time, but one last question. How do you address the social distancing and mask wearing and their compliance in the food service environment within restaurants?
We haven't addressed that with modeling at all. I think restaurants are a completely different, you know, in-service restaurants are a completely different beast. And you would, first of all, you can't eat with a mask on, and there are reasons that we close restaurants and bars, and it is because there is transmission of COVID in those settings.
Gotcha. All right, well, we're out of time, and for me, this has been just one terrific session. I really appreciate everybody's input. Kelly, Hank, Mark, and Leanne, you were all terrific. You really nailed it. So thank you. I hear all the applause in the background, so thank you very much for your contribution. With that, we'll end our session.
Yeah, I would like to thank all the speakers on behalf of ILSI. Thank you very much, Dr. Doyle, for chairing the session, and I'm inviting all participants to join our 5th session on Tuesday next week. It's a session sponsored by the American Society for Nutrition for the 2nd year in a row, so we're very happy with our partnership, and we'll hear about consideration for implementing globally harmonized nutrient reference value. We'll have speakers from the United States, from Thailand, from Ghana, and from India. So very international perspective on this very important topic. Again, thank you very much for attending today's session, and we look forward to welcoming you next week. Thank you very much.